Hello everyone, welcome to yet another session of online biology tutorials. I'm here, Dr. Nidhi Madan Chadda, the owner of these tutorials. I hope everyone has subscribed to the channel. If you haven't, please do so immediately. In this video, we will study about nucleosomes. What are these, right? In the previous video, we have already discussed about the dimensions of the bDNA. We have seen that the base pairs are 3.4 angstrom apart. And we also came to know that 10 angstrom is equals to 1 nanometer. So if we want to convert this value in nanometer, it comes out to be 0.34 nanometer. Right? So the amount of DNA which is found in a cell, it is approximately 2.2 meters, which is quite high. And DNA will not be able to accommodate such large, lengthy amount of DNA. Thus, packaging is a must which is required in DNA. Packaging means the compaction will happen. Folding will happen so that it can be easily accommodated in the nucleus. Now, packaging in the case of prokaryotes is different, and in the case of eukaryotes also it is different. If we talk specifically about board examination, then we will be talking about packaging in the eukaryotes. Means packaging in the modern cell. Means packaging in human cell, because we are also an example of eukaryotes. Plants are also an example of eukaryotes. So without further ado, let's get started with our topic, nucleosomes. Now, the nucleosome term was given by Audet et al. So it is AUDET et al. Whenever we are writing et al, it means that co-workers. Okay, so that means Audet was a name of a scientist and he never came up on his own. Rather, he worked with certain co-workers, and that is why Audet et al. They gave the term as nucleosomes. Right? So in this we will be talking about histones. We will be talking about DNA also, because packaging of DNA has to happen. Now, first of all, what are these histones? Because this is altogether a new term for you. So, histones are actually the proteins. Now, when I am using the term proteins, that means there must be amino acids, because amino acids are the monomers for forming the proteins, and proteins are the polymers. So histones are the basic proteins, and they are rich in two amino acids, which are lysine and arginine. Right? Lysine and arginine, they are in prominent amount in the histones. And both of these amino acids, they are positively charged. That means the overall charge on the histones, it is going to be positive. And the overall charge on DNA, it is negative. So DNA is negative because of the phosphate group, and histones are positively charged because of the lysine and the arginine. Now, first of all, histone octamer will be formed. Okay, octamer word when I am using, that means it would be including 8 molecules, and that is why the word octa is there, right? So 8 molecules will be there. So histone molecules like H2A, H2B, H3, H4. Now, because this is a two-dimensional view, you can only see a tetramer. But behind this, another molecule of H2A is present. That means we are using dimer. We are using 2 molecules of H2A. Similarly, we have used 2 molecules of H2B, 2 molecules of H3, 2 molecules of H4. Means ahead you can see 4 molecules, at the back also 4 molecules are there. So 4 plus 4 means 8. Now this structure, which is formed by the union of 2 molecules each of H2A, H2B, H3 and H4, it will be known as Histone Octamer because it is comprising 8 monomers, right? And of course the charge on it would be positive. Now what would happen, the DNA, suppose this is the structure, right? This is the structure of histone in my hand. Now DNA, when it will be wrapping around, it will be taking a one complete turn. Other than one turn, it will again wrap. But then in the next turn, it will cover just 75% of the histone. So one complete turn and another turn 75%. So that makes how many turns? 1.75 turns. So DNA takes 1.75 turns on the histone octamer. So as it is getting wounded, as it is getting wrapped, that means compaction is happening. Compaction is happening and folding is happening, right? So that's what we are doing it. So this DNA will be wrapped around the histone octamer. And by the wrapping of this, what would happen? If we will observe a nucleosome, it appears as this, right? So the structure, this is actually the histone octamer. And on the histone octamer, you have already understood that DNA is wrapped. But this strand is also being seen. What is it? This is the linker DNA. Okay. The DNA which is wrapped around the core or the histone octamer, it is known as the core DNA. Now you have seen that 1.75 turns of DNA are consumed while it is folding around the histone. So compaction is happening. So if we talk about core DNA and linker DNA, this makes a nucleosome. I hope this is clear to you. So many nucleosome units are being seen. See, this is one unit, this is another unit, this is the another unit, and this one is the another unit. So here the colored structures in between, they are defining you the histone octamer with co-DNA wrapped around it. So the number of turns, you have to remember, number of turns are 1.75 turns. And if they will ask you how many base pairs are used for making one nucleosome, then you would be writing 146 base pairs for the core DNA and 54 base pairs for the linker DNA. And this makes overall base pairs as 200 base pairs. So here also, if you want to show in the diagram, then you can write 54 base pairs here along with the linker DNA and 146 base pairs as a core DNA. So this topic of nucleosome, it is also very, very important. It may come for 2 marks and you are required to draw the structure. Now chromatin, it shows the appearance of these nucleosomes. So here the diagram is of the chromatin. So whenever we unwound and we open, we get the structure of nucleosome in between. They are doing the compaction of the chromatin. Now, in certain areas of chromatin, two types of chromatin are found. So you can bifurcate chromatin in two parts. One is known as heterochromatin and another is known as euchromatin. This is very important. One marker question can come wherein it may be asked what is the difference between heterochromatin and euchromatin. So one thing very important, it is transcriptionally active. That means this will help in the transcription, and transcription is the process of formation of RNA from the existing DNA. So this part of the chromatin, which is named as euchromatin, it is transcriptionally active because the folding is not so compact. So folding is not so compact in them. That is why they can unwound, enzymes can bind, and transcription will happen. Whereas if you talk about heterochromatin, it is transcriptionally inactive. Why it is transcriptionally inactive? As the name itself is making a sense that it will not participate in the transcription. Now why is it not participating in the transcription? What is the reason behind it? Because the folding is very high. So when folding is very high, it is not going to unwound. When it is not going to unwound, how could enzymes attach and they could help in the transcription process? So very important, heterochromatin and euchromatin. I hope this has been understood by you. Stay tuned for the next video. Thank you.